Hello everybody. Let me explain you how the antigens and antibody reacts. It is very important to know about how they come to close to each other, and then how they bind to each other, and then ultimately how they destroy each other. So to begin with this, I would like to show you the relationship between the infections by the pathogens and the amount of antibody produced by the host. So uh, when the host is being infected, the two things distinctly we can observe are in a primary exposure. When the host is exposed for the first time, the two immunoglobulin or the antibody, particularly IgM and IgG, these two will appear almost uh, same time. However, there is slight differences with IgGM appears little earlier than IgG. So, but however, the production is quite low. With the little productions, we will see that these pathogens would get destroyed. So, primary exposure can be easily controlled by little amount of productions of the immunoglobulin, particularly IgM and IgG. Now, if this IgG produce, and then if the person is recovered from the infection, in case if there is infection by the same pathogens in future, and that is called second exposure to the antigen. When there is a second infection, we will see both IgG and IgM start producing together at the same time. However, IgM would be much lesser amount than IgG. IgG will continue to produce in a large amount, so that these pathogen can be controlled. So these are the two differences that you need to note here. One is a primary infections, another is a secondary infection. In the primary infections, the B cells produce IgM and IgG in slightly different time, whereas in the secondary exposure, the IgM and IgG are produced at the same time, and the productions is much more higher than primary exposure. And these in the second exposure, this memory cell produce the immunoglobulin. Okay. Now moving to next is that we must understand that antigen and antibody reacts to each other like a lock and key. They are very specific to each other. Particular antigens will have the particular specific antibody. So such kind of reactions between the antigens and antibody is also called humoral uh, or the antibody mediated immune response. Humoral or antibody mediated immune response. Please keep in mind, this is very important to know. When you talk about the T cells, we talk about cell mediated immune response. Here, humoral mediated immune response or antibody mediated immune response. All right, now next is when these reactions do happens in our, the good example is our blood. When you add the B group blood to AB blood, there will be reactions, because AB blood will have different antibodies than B groups. So the each blood group will have a different antigens and different antibodies. As a result, if we mix it, we are mixing even the antibodies, and as a result, there will be serological reactions. Okay, when we say serological blood reactions, uh, these reactions and then destructions, antibody will try to destroy the foreign particles. This happens in a three steps. First thing is antigen must come close by and the antibody must react with the antigen. So there must be a binding. Second stage, once there is a binding, there must be on either agglutinations, bringing them all together, or the precipitations. The movement of all the particles will be ceased. Then final stage is neutralizations or the destructions of antigens and then antibody. Now, these antigen antibody reaction is being activated by the complement activations. There are certain proteins that activates the reaction between the reaction between the antigens and the antibody. The process in which the two targets are brought together and then it destroys. This process is called opsonization. When one of the cells becomes very vulnerable to attack by the antibody, is called uh, opsonization. For example, here antigens need to be destroyed. Therefore, in order to make antigens to be very vulnerable to the antibody, then these antibody would first attach to the antigens, and that would increase the vulnerability of the antigen. Thus, this term opsonization is very important here. All right. Now, this reaction is very specific. It follows the lock and key system. Uh, antigen and antibody binds together using two different sites. Antigen has the epitopes and antibody has the paratopes. These are the two reasons where they combined each other. uh, when they combined and then rest of the parts, when these two epitope and paratope combines together, rest of the parts of the antigen antibody are also brought together by a certain intermolecular forces like electrostatic force, hydrogen bonding, hydrophobic bonding, and the van der Waals force. These are some of the force which tries to keep them together. Because of that, when they come so close to each other, that leads to the avidity, refers to the overall capacity of the antibody to bind with the multivalent antigen. So antigen can bounded by many of the antibodies. As a result, the antigen becomes opsonized, meaning very vulnerable to destruction. Now there are four different types of reactions of antigens and antibody. First is precipitations. When antigen and antibody comes too close to each other, the paratope and epitopes helps them to bind like lock and key. That initiates the first reaction. Uh, this is what happens. Now there is an antigen and antibody, they starts binding, and then yeah, there will be clumping. Okay, there will be highest reaction here and there will be lesser. The peak would be here. Now next reaction is agglutinations. Agglutination is when there is a lot of antibodies binds many antigens, or many antigens binds to a few antibodies, and they becomes a clump together. They comes close to each other and they cannot break, and that is called clumping. So this is how the clumping is. The blood clumping is just forming a lump of molecules. then cytolysis. Then once this complex has been formed, there will be other complementary activity, complement proteins which will activate the this complex, and this is called this activated complex will slowly now determine how it will be lysed or the broken down. Finally, there is a final term is used is opsonization. Opsonization is the vulnerability or the where ability of an antibody where the pathogens are made more susceptible to lysis or breakdown. So ultimately, it is opsonizations or the opsonin molecules that helps to break the antigen. And if you are asked which is the opsonin molecule here, it is an antibody, because antibody joins to the antigens and then it results in breakage of the antigen. uh, with that, I think I'm able to cover, because you have to understand these two terms. Cytolysis is brought about by the process of opsonizations, and these opsonization results in the neutralization, meaning breakdown of the complex between the antigens and the antibody. I hope this will help you to understand how the reactions occurs. What you have to keep in mind is how antigens is being attacked by the antibodies, how the antibodies increases the opsonizations of the uh, antigens, and what are the four different types of the antigen antibody reactions. All right, that's all that you have to understand from this topic. Thank you.